Homework. All right. This is the instructions for the homework that you'll find on your canvas. And they're long because they're nice and detailed. Because there's some steps here, and I could do it in less words, but you know what? I'd lose somebody each time I did that. So let's not. Yeah. So hopefully this is more than you need. But all the same, I'm here for questions. All right. Now I'm going to go through the procedure over here so you can kind of get a look at it, and you're going to do some version similar to this, but yeah, not exactly the same. Yeah. All right, so here we go. All right, homework instructions, and then we're going to go to viral zone. That's going to be our first stop over here. And on viral zone, the group of viruses that we want is going to be the single-stranded RNA positive sense viruses, ssRNA plus. And I'm going to keep disappearing here because I'm a window in front of this one, but I'll come back. Okay, within this group, we're going to search in one box, look for it, Picorna viralis. This is a group of viruses that are so abundant, they are just everywhere all the time, yeah, right here, now, etc. And so this is the easiest group to find. All the steps I'm showing you, you can do them with any group of viruses. You can find wonderful things, but every time you look, you will find Picorna viruses, Picorna-like viruses. That's what we call these things for short. Yeah, even though that's probably longer. So anything in this box is fine. Click on one of the ones with a blue link on it, because the black links are not links. They don't go anywhere. Look, yeah, it does nothing. So we'll pick Locarno virus. Awesome. All right, now that you're here, have a look. So you've got a map. This is important. You're going to make a map like this, in this style. You don't have to have a little curvy poly A tail. That's okay. For your virus, yeah, whatever it may be. And it probably doesn't have a cap there. It probably has a thing called a VPG, viral protein genome associated. But whatever. Bad drawing or slightly inaccurate drawing for a mostly unknown virus. As you scroll down the page, the thing you want on this page is, look, there's going to be a reference strain. And you can pick any of those genera, but they will all have a reference strain. And the reference strain will have a name of a virus. So here we go. This is the virus, Asterianelopsis glacialis RNA virus. You don't have to pick that one. In fact, go pick a different one. Get your own virus. Yeah. Oh my gosh. Yeah. And then we're going to pick the proteome of that virus, which is all the proteins. But before we go away, just in case you're curious, where does this virus come from? It'll tell you the natural host down here for your virus, which is probably green algae phytoplankton from coastal waters. How nice. How nice. Okay, proteome. Let's go. All right, inside the proteome page, it's going to take you to something like this. It should be similar. Some viruses may just have one protein there. Some may have like 5 proteins. A lot of them will have 2 proteins there. Doesn't matter. It's okay. And this is going to work no matter which one of those you pick, probably. Yeah. Although, here, we're going to look for which one is the longest. So I'll put myself down here. So I'm just peeping out like a little mushroom. And we're going to look there. We got one with a length of 1614 versus 863. 1614 wins, so we're going with the top one. Here's the top one. Click the entry. Bam. It's going to take you to this page. You're going to click Format. And you can click Fast A Canonical, and bam, it's going to take you to this. And look at it. Look how amazing it is. That is the amino acid sequence for your new virus. No, actually not. It's for the virus that somebody else found that you're going to use as bait. So this is just like fishing. You want to catch a fish, you got to put a worm or something shiny on the hook, and the fish will try and bite it, and then you pull it out. In this case, we're going to use a DNA and RNA search engine called BLAST, and we're going to search for something that is similar to these letters, because these are single letter amino acid codes. So copy it. You can copy the top line or not. Actually, I'm going to leave it off this time. So there we go. It's copied. Awesome. Next stop, we're going to go to BLAST. Here we go. Here's BLAST. The link is in the instructions. You want T-BLAST-N. It's this guy right down here. There we go. T-BLAST-N. The best blast of them all. There's a search box right up here where it says enter query sequence. Bam. You paste your sequence. It's going to come out formatted like garbage like that. You don't care. That's not a problem. Yeah, this is all right. Then you got to change a couple things down here. One, this line where it says database, you're going to set it. It's going to start out saying this, nucleotide collection NRNT. You're going to hit it, go down the list to transcriptome shotgun assembly TSA. That's what you want. Then once that comes up, it's probably going to come up and the button next to organism is going to be clicked. If any of these others are clicked, click organism, because that's what we need to search for. And now you're going to enter in an organism. And I would say you don't enter in just like one kind of scientific name. I would go for a group because that increases your chances of finding something. We can only find something if somebody out there in the world has already sequenced its RNA. Then we have a chance of at least having something to search. So we'll cast a wide net here, and I'll do vascular plants. There we go. Let it auto-complete for you. So vascular plants, that's fine. And you notice it gives you a couple choices, but they have the same number here for the tax ID. They're actually the same thing. They're synonyms. So complete it. It'll give you a taxonomic identification. That's a number that only makes sense to NCBI and BLAST, and that's fine. So good, we got it. And then you're not going to mess with anything else. You're just going to hit blast. Bam, blast, go. Zoom. And you'll see a page like this. Now, if at any point during the search, this page goes away and you get a blank screen or something like that, it'll still have up here this recent results. And actually on any BLAST page, it will have this. You click recent results, give it a sec, and it'll show you, here's one I did earlier, and here's the current job, which is still running. If you want to go to these, like go to the job that's done or go to the current job, you just click on that, and since this one's still running, it takes me back to the page. So that's how you can refresh. If your browser gets confused and kind of loses track of the page, which is what will often happen at NCBI, that'll get you back. If you search on this and it hangs for like half an hour, which it can easily do, sometimes it comes back, sometimes you'll have to research. It's just a thing. I don't know. They need more tax dollars over there at the National Library of Medicine and the National Center for Biotechnology Information, which is the computers that we're using to do this search. They've been having problems lately, but it will work. Give it a couple tries and you'll get there. Okay. When you get your results will look like this. If your results have a red box up here that says, oh no, you've either exceeded your CPU time or we didn't find anything, go back and there are instructions for how to modify your search and then redo the search and you should find something. This is just a typical search. Look at all these things. Look at them all. Oh my gosh. These are all brand new viruses, but they're not labeled as viruses. They're labeled as some kind of, in this case, plant. Colobanthus is a plant. Miracaria is a plant. These are all virus sequences, most likely. So we need to narrow this down because we just only want to work on one. That's all you have to do for this assignment. You don't have to solve all the problems of the world. So we're going to pick one that's probably going to be a complete genome. One easy way to do that is to click this column where it says accession link, ACCLEN, and that'll sort them by size. So there they are, sorted by size, and it puts the little one at the top for some reason. So you go down to the bottom. Anything that is at least 8,000 long up to about 14,000 long is probably going to be a complete genome. The things that are smaller are probably fragments. Like a 4,900 is probably about half a genome. You can search any of these, but if we're looking for all the parts, you'll actually find all of the parts. There's 6 parts that we want to find, and a complete virus should have all 6 of the parts. There's no guarantee if you pull out, one that's 8,500, maybe the full virus was 13,000. Maybe you've only got part of it still. You know what? We're going fishing, and when you go fishing, you kind of never know what you're going to catch, right? Unless you're fishing in an aquarium, and why would you do that, you big weirdo? So that's fine. We can pick this Persea americana, which is an avocado. How do I know that? It's because I clicked on it earlier and searched it because I don't know my plants that well. Fine. All right, great. Now you'll see that all of these sequences are clicked right now, and it does that by default. if we hit download right now, you will download every single one of these genomes, and then you'll have to search through them and figure out which one is yours. Don't be that person. Don't do that. Click select all again. That unselects everything. Then just go down and click whichever one is your favorite one. And if you're doing this as a group, that's fine. Select different ones. And there you go. And then up here at the top, click download FASTA complete sequence. Not aligned, complete sequence. And voila, there it is. It'll call it seq dump some number. And there you go. That is the RNA sequence for a brand new virus that's probably at least mostly complete. Yeah. Oh, and it looks like mine is negative sense, because this virus should have a poly A tail, and what I see is a poly T beginning. And so if you reverse complement that, that is a poly A tail, and it's just backwards. But we don't care whether it's backwards. We can handle it either way. We're going to take this, we're going to copy it, and we're going to go to the next step. Next step is going to be ORF Finder. Open Reading Frame Finder. And so this thing is going to find all the proteins you could make, you could translate from your virus, forward or reverse orientation, whatever you got. We're not going to change any of these settings. We're just going to hit submit. And we're going to wait about 5 seconds, and it's going to appear, because ORF Finder is some good software. It's fast. Now, you may see something like this with one long protein, and the little arrows show you which direction the protein is being read in. So this is, we've basically pulled out a minus strand of the virus. The gene is in the reverse orientation, but that's okay. We don't care at this stage. Or you may see something like this. You may get something with 2 genes, and this one's in the positive strand. The real results will, one, cover almost all of the distance of the sequence here. So this is the start of your sequence, this is the end of your sequence, and you can see the big red box is covering pretty much everything. Or over here, we've got 2 giant red boxes that cover pretty much everything between them. So they're not going to overlap, and they're going to be the biggest things that are on there. Those are going to be the actual viral genes. These other things, they may or may not be translated, probably not translated in enough quantity to do anything, but occasionally you find something interesting in one of those. I would say, for now, don't worry about those. That's like a PhD research project, trying to dig into those. When you click one of these, you can click, here's our big one, or our other big one, click it, there we go. Down here, it'll say, this is the amino acid sequence in single letter amino acid codes. You're going to copy that whole thing, copy, and you're going to paste it into some kind of document. So great. Imagine you got a Word document, Google Docs, whatever, paste it in there. If you've got more than one open reading frame, in this case we have this one and this one, put them both in there. Put this one first, put that one second, because that's the order in which they occur, and take a screenshot of this screen, because this is a scale model of your entire coding sequence, or rather your entire virus from start to end. And so this will help you get a sort of a scale model, correctly proportioned, of what your virus is actually able to encode. All right, then we're going to take these proteins. If you have one protein, great, you got it. If you have more than one protein, take each of the proteins and you're going to put them in a separate window of HHpred. You're going to paste them right there. Bam. Awesome. And if you have 2 proteins, do them in separate HHpred windows. And you can run them both at the same time. Doesn't hurt anything. When you come in, you're not going to see those 2 things. You're going to see this. It'll be PDB and whatever the current version of that is. These are the places where we're going to search, and we're searching through 3-dimensional models. The 2 things you want to add to that are PFAM-A, whatever version we're on, and NCBI conserved domains, whatever version we're on. Awesome. Hit submit. Bam. And once you do, it's going to take you to some pages. And so chances are you'll get some results pages that look like this, or maybe look like this, or maybe look like those 2 things together in some order. So just looking at this, I can tell what we've got. If you have 2 proteins that are about 300 or 250 amino acids each, and then one big protein that's about 500, then this is going to be the helicase, that'll be the protease, and that'll be the polymerase. They always occur together. They always occur in this order. Helicase, a bit of a space, and then protease and polymerase smashed right next to each other. I'm going to ignore any of the ones that look like they're longer or shorter than the majority. Just focus on these guys. Now, if you want to know, well, how did you know which they are? You can mouse over them. And so mouse over, I just mean leave the cursor on top. Or you can click them and go down to them, but then you have to come back up. This is a little button that'll bring you back to the top. And so I'm looking at this, and it's saying it's a 2C protein. And it says it's a helicase, and it says it's an ATPase. And so I'm going to look on my chart on the instructions, and oh my gosh, that is the helicase. Those are words we expect with the helicase. Great, we did it. We did it, everybody. If we looked at this one, it would say, let's see, enterovirus 2C. So 2C is one of our keywords, and AAA plus ATPase and hydrolase, those are other keywords. Those also say it's a helicase. Over here, we're going to see, okay, polyprotein there, buried in the middle, protease. And sometimes it'll be called peptidase, and sometimes it'll be called 3C. This has 3C and protease, so that is the protease. And over here, we're going to pick any of these that are roughly the same length, and we've got a nucleotidal transferase, nucleotide binding, RNA polymerase, hydrolase. All of those say that this is the polymerase. And there we got polymerase in the middle, and there we got polymerase somewhere in the middle. RNA-dependent RNA polymerase right there in the middle. So there you go. That's how we know what these are. I'll take a screenshot of this page. And since I had two proteins, I put them in two separate HH-preds, and here are the results. So you'll have that group with two short things and one long thing. That's your helicase, protease, polymerase. And then somewhere, either at the beginning or at the end of your polyprotein, or maybe on a separate polyprotein like mine, you will have three things, which will be the three capsids. They'll all be about the same size, about 250 amino acids each. That's great. These are named kind of variably. Sometimes they'll call them just after the name of the virus, so that's not helpful. Let's go to the next one. And there it says jelly roll. Jelly roll is a name for a capsid protein. Down here, we got a VP2. VP2 is a name for a capsid protein. Down here, we've got, nope, no help, capsid protein. Oh, there you go, capsid protein, etc. And you'll actually find the same things, like this 3-NAP. You will probably find matching all three of these, because all the capsid proteins are basically the same thing. Here's the cricket paralysis virus capsid protein. And you'll see that come up in some of the other ones over here. There's a Zika virus capsid protein coming up twice, whatever. Yeah. So each of these is a capsid, and we just want a rough snapshot. So this is the polyprotein that we put in. There's the start, there's the end, and roughly this part from here, I'm aligning it to the edge of that, to about here, edge of that, that's going to be our first capsid, second capsid here to here, and third capsid would be roughly, well, let's see, stretcher, a little stretcher, roughly here to here, and this is roughly. And so then once you've got all that information, and you've got your ORF finder information, so this is where the proteins are and the direction they're in. This is what's inside those proteins, so this is in the big one, this is in the small one that I just showed you. Then you're going to make a map that's going to look like the map that we got over here. And you can do this with any program that can draw rectangles and make them colors. You can color it however the heck you like. If you want to put a label on the rectangles, I think that's the nicest way to do it. You can just like type over the top of them, something like that. And you save it as a little picture. And then you're going to make a report. And you're going to put a couple of things in the report. And we can scroll down and find those things. Bam. What was the virus you used for bait? Okay, fine. We wrote that down. Bam. What's the organism group you searched for in T-Blast-N? Like in this case, it was vascular plants. In your case, it can be, I don't know, Cnidaria, which is like corals and jellyfish. Those are cool. Down here, we've got organism with a bold O. Wow, okay. So this is the blast result. And so in our case, it was... here we go. Persea americana. That's the name of it. So it'll be a Latin name for some kind of thing. Phaseolus vulgaris, I think, is some kind of bean, maybe. Colobanthus, I think, is some kind of grass. Ulmus minor, I think, is maybe an elm tree, but I don't know plants. Yeah, you can look all these things up. But that's the part of the name that we need. This transcribed RNA sequence, they're all transcribed RNA sequence, and it's telling you which sort of sample number it came from. We don't care. Over here is the accession number where you can go and you can actually look at your genome. Usually those work. Occasionally NCBI has problems, so that's why we're not going through that way. We're just downloading it. But yeah. Use that. You're going to make, so that's the organism name, like where you found the virus. What's the length? So the length you'll find over here. And the length of this one is 10,162. Why? What a large virus we found, virus genome. What's the E value from the tBLASTn results? All right, sweet. That's got its own column on the results page. So let's go up here. And there's our E value column. So scroll down to the bottom, find the E value for our result, which is 1e minus 20, which is 1 times 10 to the negative 20th power, which is a very small number. This is basically the E value is your chance of error, the chance that this is not a good match. And that's a small enough chance that, yeah, I'm pretty confident this is a real good match. Fine. And then the last thing you put in there is the map of your virus, and you're going to put these all in some kind of like a Word document or something. Save them as a doc or a docx. Those are the easiest for me to deal with. And then you're going to turn it in through Canvas on the homework link. And there you go. You did it. We all did it. We all did it just now. I hope that helped. I hope that's a decent set of instructions for you. And whatever. We can talk about it. But yeah, try them. And chances are, if you run into problems, look at the next line. It's probably telling you how to solve that problem, because I do this all the time, and if you're running into a problem, it's one that I've run into before as well, and you can get around them. Yeah, there's always something. So, and occasionally NCBI just gets sad, and you just gotta walk away for half an hour. It's not gonna search anything, and you come back, give it another search, and it'll be okay. Yeah, we all have good and bad days, and that's okay. Yeah, part of life. Fine, there you go. Those are the instructions. Deal with them.